Okay, so good evening, everyone. Okay, so today we are in the week 6 tutorial session of the NPTEL course Genetic Engineering, Theory and Application. So I hope all of you have gone through the week 6 lecture videos. So can anyone summarize what were the topics that were covered in week 6? Okay, so till now we had what all we had seen is we learned how we can select a target gene, how we can screen for a target gene, and then once we have the target gene, then how we can clone this gene of interest in a vector. So what are the different types of vectors? It will depend on the size of our insert, right? So and our gene of interest as well. So if it is a bacterial gene that we want to clone and express, then we can use a bacterial plasmid vector. While if we want to clone a very large human gene of human gene, and we want to express that particular protein of that human gene, uh, and say for example that protein that protein has a lot of post-translational modifications and um, it requires a certain um, a specific type of folding. So in that case, we cannot clone that gene in bacterial plasmid vector, right? We have to use other vector system. So we learned about the different vector systems. So starting from what are the types of bacterial vectors, then yeast vector. If we want to clone our gene of interest in an yeast vector, so what are the different vectors available then for expression in a mammalian host? We have to clone our gene of interest in a mammalian vector. So there are different types of vectors available. Okay, so now once we have the gene cloned in the vector, then how we can check whether whether the gene has been cloned correctly or not. So what are the different methods we can use? We also learned about how we can screen whether the host cell has taken up the cloned gene of interest or it has taken up a self-ligated vector. Okay, so what are the different methods? We learned all about those. So in this week, what we learned is now once we have we are sure that our we have cloned the gene of interest in a vector, then the next step would be to express that particular gene, isn't it? So let's start today's session. I'll share slides for today. Okay, so I hope the screen is visible. So yeah. Hello everyone, I'm Akash Jyoti Pathak and I'm one of the PMRF TAs for this NPTEL course, Genetic Engineering Theory and Application. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. So let's first have a quick recap of what all topics were covered in week 6. So as I was explaining that once we are confirmed that yes, I have or my gene of interest is cloned in the has been cloned in the vector. Okay, so we have the vector, and we have a gene of interest. So now we have done sequencing, and also we have done, say, for example, we have done restriction, digestion, and PCR amplification, and we are now confirmed that yes, my gene of interest, I have the my cloned gene of interest. The next step would be what? Next step would be to, and also what we have done, we have transformed this clone gene of interest in a suitable host cell. Okay, host cell. Now the next step would be to express this gene of interest. Okay, what do you mean by expression of the gene of interest? So expression means that what? From your DNA, you have this DNA, you have your gene of interest means what? You have the DNA. Then from this DNA, what will happen? Transcription will happen, right? You will get an mRNA by the process of transcription, and then there will be translation, isn't it? So that you can get your protein. Okay. So this is called the recombinant protein. This is the recombinant protein. So now the purpose is so in the earlier sessions we talked about the different vector system, how we can screen for whether our host cell has the clone gene of interest. We have seen all that. Now the next step is to check the express this gene of interest in the host cell. Now this clone gene of interest can be transformed in different host cells. So what are the different host cells that can be used? These are the bacterial host cell. It can be the yeast host cell. It can be the insect cell line, or it can be mammalian cells. Okay, so depending on where you have transformed your clone gene of interest, in which host you have transformed your clone gene of interest, there are different ways of expressing the uh, protein from that gene of interest. Okay, so we will see one by one today that that how we can express the protein in these different host cells. Okay. Okay, so let's first learn about, so we all know about the protein synthesis machinery, isn't it? So in the basic molecular biology, if we have a little bit knowledge about basic molecular biology, then the central dogma of molecular biology, what does it say? That from DNA, the information is passed on to mRNA, and from mRNA, we we get the protein by the, so this process is called transcription, and then from mRNA to protein, that process is called translation, isn't it? Okay. uh, so uh, so to so uh, so for synthesis of our protein, what will be required? So for expression of the DNA, what all things must be present in the vector, that we will see today. Okay, so the protein synthesis is a multi-step process. It begins with the binding of the RNA polymerase to a promoter to start transcription. So your gene of interest, so you know about the transcriptional unit, right? So if you have this gene of interest, so your gene of interest must be flanked by a promoter and a terminator, isn't it? We have learned all this in class 12 as well. So it must be flanked by a promoter, and it must be flanked by a terminator. Okay. So how will this gene of interest be expressed? So first of all, what happens? The RNA polymerase. The RNA polymerase will bind at the promoter region, right? RNA polymerase will bind at the promoter region. Also, this is the sequence which must be present in your DNA, isn't it? So your DNA must have a promoter, it must have the gene of interest, and it must have a terminator. So first of all, what happens? This is your RNA polymerase. It has a C-terminal tail. This RNA polymerase will bind to the promoter, and then uh, once it has recognized the promoter, then there will be a transcriptional start site. So at the transcriptional start site, your DNA will get transcribed to mRNA, isn't it? It will get transcribed to mRNA, right? And then what will happen? Once it has reached the RNA polymerase has reached the terminated sequence of the DNA, it will fall off from the DNA sequence. So it will get removed uh, from this DNA, and And so this process takes multiple times. So depending on what is the level of protein that is required by the cell, this transcription can take place multiple time, and as a result, you will get multiple mRNA transcript. You will get multiple mRNA transcript, and then once you have the mRNA transcript, then what happens in this mRNA? Your ribosome will come. Your ribosome will attach to the, so there is a ribosomal binding site which is present on the mRNA, and the ribosome will come and then it will translate this mRNA into protein. Okay. Okay. So the first process is what? It begins with binding of RNA polymerase to a promoter to start transcription, which forms messenger mRNA or the messenger RNA. Once the mRNA is synthesized, the translational machinery begins the synthesis of polypeptide, which is the protein. So that is what has been explained here. So first your RNA polymerase will bind at the promoter, and then it will start transcribing your gene of interest from the transcription start site, and you will get an mRNA. Once your mRNA is formed, then the ribosome will come, then there are tRNA. So all these different molecules will bind to your mRNA, and it will start synthesizing the amino acids. Depending on what is the codon which is present in your mRNA, you will get your amino acids will be attached one by one, and you will get a polypeptide chain. Okay, so what one thing to note is that in bacteria, the transcription and translation can occur simultaneously, with translation starting as soon as the mRNA emerges from the RNA polymerase. So protein synthesis typically starts at a start codon and ends at a stop codon. Okay, so now what is explained over here is that so once your so first of all, what will happen? Your RNA polymerase will come and bind to your promoter region. Okay, then in the transcription start site, your transcription start site will have the sequence. So if it is a DNA, it will be what? It will be ATG. Okay, and from here you get the, once it is transcribed, you get this AUG. Okay, so this is your start codon. Start codon is your AUG. So this is with, when we talk about codon, so, so when we talk about codon, we are saying everything with respect to the mRNA. Okay, so basic transcription concept you all know, right? Only one strand of the DNA can be used for transcription. Both the strands cannot be used. The strand which is used for transcription, that is called the non-coding strand, your template strand. So these all things you must be knowing, right? Okay. So now let's see this is figure, and let's understand what is this transcriptional unit that I was talking about. Okay, so this is your DNA. So your DNA, the transcriptional unit of a DNA will have a promoter. It will have a it will have a coding region. So this is called the coding region or structural region of the DNA. And this is your terminator. So first your RNA polymerase will bind to the promoter, and it will start transcribing the gene from your transcription start site. Okay. And and it will stop at the termination site. Okay. So you get a, so this is the mRNA that has been synthesized. So this is your 5 prime. And what happens during transcription? Only one of the strand is used for transcription. Okay, because if both the strands are used, then the mRNA that is produced, they will be complementary to each other, and then your protein cannot be synthesized from this RNA. Okay, so only one of the strand is used. Now, this is your mRNA. So when we are talking about mRNA, this mRNA has a, so when your ribosome will come and translate this mRNA, then what happens? It has a 5 prime untranslated region, which means that your protein will start synthesizing from this region of the mRNA. Okay, so this is the start codon, which means that this start codon will have, what is this sequence which is present in start codon? It is AUG. Okay, and this is your untranslated region. What does your untranslated region have? Your untranslated region has the ribosomal binding site. So this is the region where your ribosome will come and bind. Okay, so you have the 50s and the 30s subunit of ribosome. So it will come and bind at the 5' untranslated region. So this ribosomal binding site in prokaryotes is called the Shine-Dalgarno sequence. So your 5' untranslated. So this is this yellow colored region that you see in the mRNA. That is your ribosomal binding site, and this part is not translated into protein. Your protein translation will start from this particular region, from the start codon, and it will stop at the this stop codon. So your start codon is AUG, and your stop codons are what? There are three stop codons: UAA, UAG, and UGA, isn't it? Uh, okay. And then you have a 3' untranslated region. So this was the uh, this is the basic knowledge of molecular biology that you need for understanding that how these constructs that we transfect in different host cell. What must be present in this construct? So it must have a ribosomal binding site. It must have a promoter region. It must have a terminator site. So all these regions must be present in your construct, so that when you're transfecting this your this construct or your this cloned plasmid into your host cell, then your gene of interest will be expressed. If these regions are not present in your construct, then your gene of interest will not be expressed. You will not get your protein. So you must have this basic knowledge of molecular biology that yes, these sequences must be present. Okay. So what are the essential components for protein expression? First is your promoter. Okay. So what is the promoter? So promoter is that region in your DNA where the RNA polymerase will bind and it will start transcribing and start transcription, initial transcription. So this is the sequence upstream of the gene that acts as the docking site. Docking site means what? Here your RNA polymerase will bind and then it will initiate transcription. Okay, so initiation will start from the transcription start site, but this promoter region must be present so that this sequence will specify that which is my portion of the DNA which has to be expressed. So this region must be present. Okay. Then there is the ribosome binding site. This sequence, which includes the Shine-Dalgarno sequence, provides a docking site for the assembly of ribosomes. Okay. So one thing you must understand is that the DNA, you do not have the RNA. The plasmid that you have, that is a double-stranded DNA molecule. Okay. So your DNA sequence must have your promoter sequence. There must be a promoter sequence in your DNA. There must be a ribosomal binding site. So somewhere here you must have your ribosomal binding site, or if it is a bacterial expression host, then on a bacterial vector there will be a ribosomal binding site host the promoter region. Then after that there will be your gene of interest, and there will be the sequence of your gene of interest, and then following your gene of interest there will be a transcription termination site. Okay, and after that you can add an affinity tag. So what is an affinity tag? We will learn today. So why is it required? So there can be different types of affinity tags. There can be 6S or 6 histidine tags, which means that there will be the um sequence of the DNA which will code for histidine. Okay, so six which will code for 6 histidine molecules. Okay, so why histidine molecules? So it will help in this affinity purification, nickel anti-affinity purification. So we will learn that. So now remember that. So let's see. So this was covered in the lecture as well. So first you must have your promoter, then there must be a ribosomal binding site, then there must be the sequence of your gene of interest, then there must be termination. So what will happen once you have once you have transfected this your construct in the host cell, then your RNA polymerase will come and bind to the promoter region, and then what will happen? Then your gene of interest will start getting transcribed. Now in the next step, what will happen? Once you have the mRNA, there will be the ribosomal binding site, which will be present on the mRNA. So your ribosome will come and it will start translating it into protein. Okay. So that is what is explained here. So binding of RNA polymerase to the promoter elements to start transcription to form mRNA. Now, as soon as mRNA is synthesized, translational machinery. So what is the translation machinery? Those are the ribosomes, tRNA, isn't it? And the mRNA. Together, all these will be the translation and machinery will come, and the protein synthesis starts usually at the start codon. Remember this. What is the start codon? It is AUG. AUG codes for methionine, and ends at the stop codon UAA, UGA, or UAG. Okay, so these three codons do not code for any amino acid. In bacteria, transcription and translation occur simultaneously, and translation starts as soon. These all things you know. If you have taken biology in our class 12, then you must be knowing this. So, because this bacterial, there is no compartmentalization, isn't it? So there is no separate nuclear envelope, and so your nucleoid is lying freely in the cytoplasm. So what happens as soon as this mRNA starts getting transcribed, the ribosome also will be so at one site, your RNA polymerase is transcribing your gene, your mRNA is getting formed, and at the other end, your ribosome will come and bind to this mRNA, and it will start synthesizing your protein molecules. Okay, so amino acid 1, amino acid 2, and you will get a polypeptide chain. But in this eukaryotic system, the mRNA that is produced, that requires some processing, isn't it? Your introns will have to be removed, and then this cap region and tail region, all these needs to be synthesized. And after this different modification, your mRNA comes out from the nucleus into the cytoplasm, and then in the cytoplasm you have the ribosome, and then translation can take place. Okay, so now, when you are choosing an expression, so that was all like what all should be present in your in the vector, or in the construct that you design. So you have to design a construct where you should have all these promoter region, ribosomal binding site, your gene of interest, your terminator, all these things must be present. Now, once you have designed your construct, then the next step is what? You have to transfect, or you have to insert this gene of interest into what? Into a host cell. So how do you choose that what should be the host? There are different host cells that are available. There are bacterial. So you can transform, or you can put your clone gene of interest inside bacteria. You can put it inside yeast. You can put it inside mammalian cells. You can put it inside insect cells. Okay. So how will you decide which insect? Which host cell to use? How do you decide this? So that will depend on the size of protein. So if it is a very large protein, then so for example, small cytosolic proteins and polypeptides that are less than 100 residues, they can be expressed in E. coli. Okay, so but cytosolic proteins that are greater than 100 residues, which means that they are having more than 100 amino acids, they are difficult to express in E. coli. And so what happens if you try to express very large proteins in bacteria? Then this bacteria treats this protein as something foreign, and it packs them in certain vesicles. These vesicles are called inclusion bodies. Okay, and these inclusion bodies are they treat these proteins like garbage. They some garbage, and they put it pack it in some vesicles, and they store it in the periplasm. Okay, secrete it in the periplasm. Okay, the protein the protein which is present in your inclusion bodies, those proteins are not properly folded. They are misfolded, and yeah, and if it is misfolded, then there is no purpose of this protein. You cannot do any further studies with this protein, because your protein is misfolded, and your protein will not perform the desired function. Okay, the protein must if it is it should have the native structure. Okay, so that is why it will depend on the size of the protein. So if it is a small protein which is less than 100 residues, then you can express that protein in bacteria. But if it is having more than 100 residues, then the such type of protein gets are packed in these inclusion bodies, and they are misfolded. So you cannot use bacterial. So you can obviously. So you can obviously purify the protein from inclusion bodies as well. So in that case, you have to first denature the protein. You have to treat it with urea, guanidinium hydrochloride. You unfold the protein, and then you purify the protein. And after your protein is purified, then you refold the protein by dialyzing it. Okay. Now, what is the amount of protein that is needed? That will also determine that what expression system I have to use. So if small quantities of target protein is required, it is not required to optimize protein production. But if large quantity of protein is required, then we have to try different host vector systems. What will be the expression of your protein? That will be that is decided by what is the type of host cell you are using. So, for example, if you are trying to express any human protein in a bacterial system, then you will not get very high yield of the protein. If you are trying to express a bacterial protein in a bacterial host, then then the yield of that protein will be very high. You are setting up a culture of 200 ml, and in that two from that expressing the protein from the 200 ml culture would be enough to perform all the experiments. But if you're trying to express some human protein in bacterial cell, then sometimes it becomes difficult. So it depends. All of this depends on the property of the protein. What is the protein that you want to express? So now whether active protein is required, that will also depend. So if the protein will be used in biochemical or cell biological studies, then it is important to obtain soluble or active protein. So that is what I was explaining. So what happens is that when your protein gets, so if your protein is present in your inclusion bodies, then this inclusion bodies always comes in your pellet fraction. So what happens is that, so uh, so 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 if you are expressing a protein in a bacteria, for example, so your bacteria, so what you do is, let's explain this. Okay, so now let's understand this. What happens? So you have your, so for example, you are trying to express a protein in a bacterial host. Okay, so you have this is usually what we do when we are trying to express a protein, we take PET vector. So there are different types of PET vector that is available commercially, PET 28A, PET 28B, PET 11A, PET 3A. So there are series of PET vector that is available. Now what you have done, you have cloned, this is your gene of interest, and you have cloned your gene of interest in your PET vector. Okay, so now you did all these studies to be assured that, yes, your gene of interest is present in this vector. You did sequencing and you are confirmed that yes, I have cloned my gene of interest. Now next step, what you do is you transform your bacterial. You take this back. You take the bacterial cell. You make it competent by treating it with what? Last class we were discussing how to make competent cells by treating it with divalent cations like calcium chloride. And now you have, so this is your bacterial host, and then you have put your cloned gene of interest in this bacterial host. Okay. Now, in the next step, what you want to do? Now, in the next step, you want to express this gene of interest. So, what you do? You First of all, what we do is we take a we inoculate the. So, first of all, actually, what is done is, so we have transformed the bacterial cell with the clone gene of interest. And then we have plated it. On a agar plate, we have plated it. And then we have got several colonies of the bacteria. Now, in the next step, we take one colony and we inoculate it in a 10 ml culture, in a 10 ml LB media. So this is my 10 ml LB media, and then we inoculate this. We pick up one colony and we inoculate it in this 10 ml media. Then so this is called the primary culture. And then in the next step, what we do, we take this primary culture and then after, so we keep it for, say, for 16 hours. We allow this the bacteria to grow. We allow the bacteria to grow in this culture, in this media, and then from this media we take around 1% of this we take, and then we inoculate it in a large culture. Okay, so this is your 750 ml. For example, this is your 750 ml LB media, and then we inoculate this. We take 100 microliter and we inoculate this in, or not 100 microliter, we take 7.5 ml, and so 1% of this is inoculated in your LB media, and then we allow the bacteria to grow. So this is called your secondary culture. Okay, and then after the secondary culture, what we done after a particular OD is reached. So that again you have to optimize that what is the OD that you want. So after you have reached a OD of say zero point 0.4 to zero point 0.6, then we induce it with IPTG. Okay, we induce it with IPTG. Now once you induce it with the IPTG, so once you induce it with IPTG, your protein starts getting expressed. Now your bacteria will start expressing the protein. Now there can be two things. First is that either your protein can be, now it will express the protein. Okay, protein is expressed. So after yeah, this entire thing is done, so you harvest this bacteria, harvest the bacteria, which means that you centrifuge your culture. You centrifuge your culture and you will get the bacterial pellet. Now this bacterial pellet is sonicated. Okay, is sonicated. So you have to rupture the bacterial cell wall. So because your protein is expressed in the inside the bacterial cell, you will do some mechanical disruption or chemical disruption. There are different disruption methods. So most commonly, a sonication is done, and then once you sonicate it, there can be either your protein can be expressed in the. So after you have sonicated it, then again you spin it. Once you spin it, you get two fractions: your pellet fraction and you get the supernatant. So if your protein has been expressed in the inclusion bodies, then your protein will be present in the pellet. Okay. But if your protein is soluble, if your protein is soluble, then you will get your protein in the supernatant fraction. So you have to check whether your protein is getting expressed in the inclusion bodies or whether it is soluble and it is staying in the supernatant. So if it is remaining in the supernatant, then it is easy. Purifying that protein is quite easy. But if it is your protein is getting is present in the inclusion bodies, then there are other steps which you have to follow. You need to solubilize the inclusion bodies, and then you have to you have to denature the protein because whenever a protein is expressed in inclusion bodies, yeah, the protein is misfolded. It is not, it does not have its native structure. Okay, so you need to unfold the protein, denature the protein, and do all the purification steps. And after that, you will again refold it by dialysis. So this is the crux of what how your protein is expressed in a bacterial host. Okay, so what are the criteria to select a expression system? So this is what I have taken from the lecture slide. So what is the quantity of the desired protein? So that is what I was explaining. So if a protein is required in small amount, any host expression system may be suitable for the purpose. But if a large quantity of the protein is required, an E. coli, yeast, or bacterial expression system might be more suitable than mammalian expression system. Okay. So depending on what is required, if you want to do some biophysical experiments, for example, you want to study the structure of a protein, you want to do determine the structure by X-ray crystallography or NMR or cryo-EM, then in those cases, you will require a very high amount of protein. Okay, around 1 milligram of protein will be required. So, but if you do want to do some biochemical studies uh, or just you want to see the level of expression. So in that case, yeah, you will not need much high amount of protein. Okay, mainly for doing biophysical experiments, you require a very high amount of protein, pure protein, and also that will depend on the size of protein. So if you're that is what I was explaining. If the size of the protein is less, then it can be expressed in E. coli. If it is very high, then your bacteria uh, will uh, will create inclusion bodies and it will be insoluble. It will go in an insoluble fraction. And also it will depend on the compatibility between the source organism and the expression system. So what is that gene, whether it is a human gene, whether it is a viral gene, for example, you want to study the viral protein, the structure of some viral protein. So the depending on all this, your yield of protein will depend. Okay, so in general, a close distance between source organism and expression system. So that is what I was explaining. So if you have a bacterial protein, then you express it in a bacterial system, in a bacterial host, you will get a very high yield of protein. But if, for example, if it is a viral protein and you are expressing it in a bacterial host, then the amount of protein will be less. And also it will depend on the downstream application. So that is what I was explaining, that if you want to do some determine the structure of protein, you want to do NMR, cryo-EM, X-ray, crystallography experiments, you will need very high amount of protein. Okay, but for if the protein production is for generating antibodies, then also you will require very low amount of protein, right? Any expression system may suit well. Okay. Okay, so I hope all these points are clear. So if you are not able to understand any point, then you can ask me. Okay. So now answer this question. The start codon for protein synthesis in most organisms is what?
D.
Yeah, what it is?
D. AUG.
AUG, right? Correct. Okay. So AUG is your start codon and it codes for what? It codes for methionine. Okay. And what is your stop codon? You also remember the name of the stop codon: UAA, UAG, and UGA. Okay. Okay. So yeah. So what is a promoter? Let's understand quickly. So promoter is that region of your DNA where your RNA polymerase will bind and it will start transcribing. And this promoter is that sequence which is present upstream of your gene of interest. So if this is your gene of interest where transcription has to take place, this is the upstream region of your gene of interest. This region is called the promoter. Now this promoter region has certain consensus sequence. So this is the promoter region of a bacteria. So different hosts will have different promoters. Okay. So a bacterial promoter region has the two consensus sequence. What do you mean by consensus sequence, anyone? So, so consensus sequence basically means that this sequence, if your DNA has this sequence, then this is conserved to some extent, which means that this sequence, that is TATAAT, this will be present in most of the bacteria. It will have some minor variations. It will not be exactly the same in all the bacterial promoters, but mostly it will have this TA-rich sequence. So this is called this region is called the Pribno box, which is present. So how is this? So this how is this transcriptional unit? Their nomenclature. So the start region is designated as +1, and upstream of the transcription start site, everything is numbered as minus. Okay, so upstream it's -1, -2, -3, like this it will go. Okay, and this -10 region, you have this Pribno box, and in this -35 region you have the Tata box. Okay. So these are the particular sequences which defines a promoter region. So your RNA polymerase will look for these sequences and it will identify that, yes, this is my promoter region. And now I have to start transcription from post 35 residues from this sequence. Okay. So the promoter are the DNA sequences that are recognized by the transcription apparatus and are required for transcription to take place. So in bacterial cells, promoters are usually adjacent to an RNA coding sequences. So upstream of the RNA coding sequence, you have the promoter and essential information for transcription, where it will start transcribing which strand is to be read and in what direction the RNA polymerase will move. The most commonly encountered consensus sequences found in almost all bacterial promoters is centered about 10 base pair upstream of the start site. That is why it is named as 10 minus 10, and which is present 35 base pairs upstream of the transcription start site. That is called the -35 consensus sequence. Okay, so that is called so your -10 consensus sequence is called the Pribno box, and its consensus sequence is TATAAT. What does it mean? Consensus sequence means that this is conserved in most of the bacteria, which means that all these bacteria will not have this exact sequence TATAAT, but it will be similar to this sequence TATAAT. Okay, so another consensus sequence common to most bacterial promoter is TTGACA, which lies approximately 35 nucleotides upstream of the start site and is termed the -35 consensus sequence. Okay, so all this you must have learned in class 12, isn't it? Promoter region, what are this -10 consensus sequence, what is the -35 consensus sequence, Tata box, Pribno box. Okay. So the sequence essential for RNA polymerase docking in an E. coli expression vector is what?
Promoter.
Yeah, promoter. Correct. Promoter. So yeah, so this is again from the lecture slides. So it must have a promoter. Promoter is the docking site. Ribosome binding site is what? So the ribosome binding site is the Steindl-Garno sequence, which is present in bacteria, or it is the Kozak sequence, which is present in eukaryotes. You all know about all these basic molecular biology, isn't it? So, and then there is a termination site and affinity tag. Okay, so all these things must be present in your expression vector. So if you want your gene of interest to get expressed into protein, then you must have these sequences present in your construct. Otherwise, your gene will not get expressed. So the result of substituting nucleotides in the promoter region at -10 and -35 positions, what will it result in? Option A is increased ribosome binding. Option B, enhanced protein folding, reduced mRNA stability, altered transcription initiation event. So so your -10 and -35 position sequences are they are what? They are the promoter or the consensus sequences which are present in the promoter region of the DNA. And it is at this region where your RNA polymerase will bind and it will recognize um, the transcription machinery, okay? Or the this transcription unit will be recognized at this position by the RNA polymerase. So if there is any mutation in this -10 consensus sequence and -35 consensus sequence, then what will happen? Then your transcriptional initiation will not take place, right? Your transcription initiation or your transcription of the mRNA will be uh, 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 disrupted, or you will get less expression of the mRNA. Okay, so altered transcription initiation events is the answer. Isn't it? So you see, this is the RNA start site. So at this region, your mRNA will be transcribed from this region. But upstream of that, you have this promoter region. And there is a -10 region, which is called the Pribno box. And there is a -35 region. So the sequence at the -10 and -35 are crucial to facilitate RNA polymerase and consequently determine the strength of the promoter. Okay, so the stronger the binding of the RNA polymerase to the promoter, there will be more expression of the protein. Okay, so the nucleotide substitution in this region severely affects the turnover of RNA polymerase binding events. Okay, so if there is any alteration in the sequences, first of all, it will your RNA polymerase will not come and will not bind so nicely to your promoter region as it was able to if it was not mutated. So that will affect the binding of the RNA polymerase to the promoter region. And what else can can occur? It can, as a result, it can also alter the transcription initiation event. So you will get less mRNA transcript, okay, or less protein expression as a result. Your binding of the RNA polymerase to the promoter will also get hampered. So you can read all this. So the function of these consequences, consensus sequence in bacterial promoters have been studying by inducing mutations. So how did the scientists or the researchers come to know that these sequences are very necessary? So they did several mutations in these sequences, and they found that if we are mutating this, the A, for example, you mutate this A to T, then this now what will happen? Your RNA polymerase is not able to bind. You are getting less expression of the mRNA. So this is how it was identified that these regions are very essential for RNA polymerase binding to the promoter region. So the results of these studies reveal that most base substitution within the -10 and -35 consensus sequences reduce the rate of transcription, which means that less amount of mRNA will be formed. These substitutions are termed down mutations because they slow down the rate of transcription. Now, occasionally, what can happen? A certain mutation can increase the rate of transcription. So, such an change is called an up mutation. Okay. So, your transcription initiation events and your expression of the mRNA will be determined by what is the what type of sequence you have in the promoter region. Now, expression vectors containing an IPTG inducible promoter. So I was explaining that how you induced that bacterial cell to produce protein. Okay. So there are different mechanisms by which this induction can be done. So one is the most commonly used in most labs is the IPTG induction. Okay, so what is IPTG basically? So you all must have learned about this lac operon in class 12. So So this IPTG is actually an analog of what? It is an analog of lactose. So if you remember a little bit about lac-operon, then what is this lac-operon which is present in bacteria? So you have a promoter sequence, then there is an operator, right? So you have a promoter sequence, then you have an operator, and then you have the structural gene. Okay. So prokaryotes have polycistronic and eukaryotes have monocistronic, all these things you have learned, right? And then you have the terminator sequences. So what happens is that, so this at this operator sequence, the repressor protein binds. Okay. You have the repressor protein. So once this repressor protein is bound to the operator sequence, then your RNA polymerase will not be able to transcribe the gene of interest. Okay. But once you add lactose to your media. Once you add lactose to your media, then what will happen? This lactose will go and bind to your repressor. There will be some conformation change in the repressor, and now this repressor will no longer be able to bind to your operator. Okay, so once this repressor is removed, then your RNA polymerase can come and can transcribe this gene of interest, and it can synthesize the mRNA. So that is your lac-operon, isn't it? In very simple terms. So there are a lot of other things, but in a simple terms, this is what is happening. So this analog, there is an analog of lactose. Analog means what which is present is IPTG. So IPTG is an analog of lactose means what? If you add this IPTG, then this IPTG can also go and bind to your repressor protein, and this repressor protein can be removed and expression can start. So why does this system exist? So if that this system exists, that if a particular protein is not required, then there is no need of transcribing that protein. Why? Because transcription is an energy requiring process. You require ATP and this entire machinery, all these different enzymes, they all require ATP, and different types of modifications have to take place. So this is an energy intensive process. So if you do not require the expression of a protein, then the expression of that gene is turned off. Okay, so that is why such system exists in bacteria. So we are using the same system when we are expressing our protein. So what happens? So this is IPTG. Remember that is an analog of lactose. And so there are different types of promoter. One is this TRP lac promoter. Then there is this TRP lac promoter TRC, and there is a lac promoter. Okay. Simply. Okay, so what is this TRP lac promoter? This is also called the TAC promoter. So a hybrid promoter containing the -35 region from the TRP promoter and -10 region from the lac UV5 promoter. It is regulated by the lac repressor and is independent of the CAMP regulation. So what is the example plasmid? This is PKK2233 is a commonly used TAC promoter plasmid. So by say, different types of these promoters have been engineered. To so there are a lot of experiments have been carried out to see that which of the promoter is causing more expression of my protein, which is causing less expression of the protein. So now another hybrid promoter similar to TAC, but the only difference is the distance between the -35 and -10 region. So 17 base pair in TRC, 16 base pair in TAC. Okay. So between this TRP, so you see there is a TRP promoter and there is a LAC UB5 promoter which has been fused together, which is giving us the TAC promoter. Okay, but the distance in this TRC promoter, the only you also in TRC promoter also you have the TRP promoter and the LAC UB5 promoter, but the distance between these two promoters is less. In so it is more. So it is 17 base pair is the distance between this TRP and LAC, and 16 base pair is the difference between this TRP and the lac UB5 promoter in this stack promoter. Okay, so remember these things are something you have to remember, right? PTRC99A is an example, and the most commonly used is the lac promoter. Okay. So a lag promoter is used in the general purpose. So most of these vectors, the PET vector that we use, they have this lag promoter often for blue-white screening. So the PUC vector, PTZ vector, PSK vector, blue script vector, PGEM vector, all these have this lag promoter. So it is weaker than the TAC or TRC promoter, but still enables expression at respectable levels. And maximum induction requires activation of the cyclic AMP activator protein, which is most active when cells use a carbon source other than glucose. So now I will okay. Let's first discuss this thing, okay? So how so there are different types of host cells that are used in the lab. Okay, so this bacterial host cell, which is widely used in any type of protein biology lab is what? So there are two types of host. One is there is an expression host and then there is a um, what you can say it is a cloning host. So there are different types of expression host E. coli that are available. So these are like you have this BL21DE3 cells, then you have this Rosetta cells, then there are BL21 RIPL cell, BL21 RIL cells. So these are the expression host. But if it is a cloning host, which means that there will be no expression of that protein. So you have the DH5 alpha. Okay, DH5 alpha, then there is an XL host where what happens only your plasmid will be maintained in this host, but there will be no expression of the plasmid. Okay, or expression of the gene of interest. But in an expression host, it will have all these different machinery that will allow the expression of the gene of interest. So what is done is basically, so this is the IPTG
Sir,
inducer. Yeah.
could you, could you please just go slight slowly in explaining?
Okay. Okay.
Thank you.
So look at this figure. So if you want to express your gene of interest means what? You want to produce the protein from your gene. So what is done? Your plasmid, your cloned plasmid is transformed in a expression host. So there are different types of expression hosts which is available in the laboratory. Okay, so these are called the BL21DE3 strain of E. coli. Then there is Rosetta strain of E. coli. So when you are working in lab, you will get accustomed to all these different host cells that are used. So why these type of host cells are able to express your gene of interest? So let's understand that thing. Okay. So these are engineered, these E. coli have been engineered. So how they have been engineered is that you see this is your E. coli genome. So look at this figure, you have this E. coli genome. Now in this E. coli genome, there is a LAC-I gene. Okay, so by engineering, you have put this LAC-I gene in this E. coli genome, and this DE3, there is another gene, this is DE3. So your DE3 gene has your LAC promoter and a LAC operator. Okay, and then downstream to your LAC promoter, you have the T7 gene. Okay, so what happens is that this LAC-I gene will code for a LAC repressor. Okay, it will code for a LAC repressor. And what does a repressor do? Your repressor will go and bind to your operator. Okay, so your repressor will go and bind to the LAC operator sequence, and there will be no expression of this gene of interest. Okay, now so you see this is the E. coli RNA polymerase. So this E. coli RNA polymerase is no longer able to synthesize this gene. So what is done is basically you have this T7 RNA polymerase gene. So you have the gene for T7 RNA polymerase, which is cloned downstream to this LAC promoter gene. So when this LAC repressor is there, your T7 RNA polymerase will not be expressed. Okay. Now, this is your plasmid. This is the plasmid where you have this is your pegged vector plasmid where you have cloned your gene of interest. See your plasmid also has this LAC-I gene, and it will have your gene of interest. So your gene of interest is cloned downstream to the T7 promoter. So now since your RNA T7 RNA polymerase is not expressed, this your target gene will also not be expressed. Now, when you add IPTG, what will happen? Your IPTG will go and bind to the LAC repressor. Okay, what I'll do is I will draw and make you understand this. Okay, so this is a expression host. So how your expression host is able to synthesize or is able to produce the recombinant protein. Okay, so your expression host. So if you see, this is your bacteria. Okay, so this is your bacteria. Now, what will a bacteria have? It will have its own genome. Okay, so this is its own genome. This is the bacterial genome. Okay, this is the bacterial genome. And now you have transformed this bacteria. Okay, so your bacteria will also have the plasmid, the cloned plasmid. This is your clone plasmid. It will also have the clone plasmid. Okay, so since this is a bacterial host, we have cloned the gene of interest in a pet vector. So this is the pet vector. Okay, so now this expression host has been engineered in a way that this bacterial genome. So if we look at the bacterial genome, then this bacterial genome has a LAC-I. It has a LAC-I gene. Okay, it has a LAC-I gene, and then there is a LAC UV5. There is a LAC UV5 promoter. Okay, there is a LAC UV5 promoter, and then you have the T7. So there is an operator as well. There is an operator sequence, and there is a T7 RNA polymerase gene. You have the T7 RNA polymerase gene. So these things are present in the bacterial genome. Okay, do not get confused. So what your LAC-I gene will do? Your LAC-I gene will synthesize your LAC-I repressor. Okay. So what now? Once this LAC-I repressor is synthesized, your LAC-I repressor will go and bind to the operator, isn't it? It will go and bind to the operator, and it will not allow this T7 RNA polymerase from getting transcribed. So here there will be no expression of this T7 RNA polymerase. Okay, so this is your T7 RNA polymerase, and your T7 RNA polymerase is not getting expressed. Now in this PET vector, now we will look at the structure of the PET vector. So if you look at the PET vector, so the PET vector is designed in such a manner that you have the T7 promoter. So there will be a T7 promoter. Okay, there will be a T7 promoter, and downstream to your T7 promoter, you have your gene of interest. Okay, you have your gene of interest, and then there will be the terminator. Okay, so now since your T7 RNA polymerase is not getting transcribed, then so your gene of interest will also not get transcribed, isn't it? Because if you are not having this T7 RNA polymerase, then there will be then this T7 RNA polymerase cannot bind. Since you do not have the T7 RNA polymerase, it will not go and bind to the T7 promoter, and your gene will not get expressed. Now, once you add IPTG, so once you add IPTG, what will happen? This IPTG will first go and bind to your this LAC repressor. Okay, so once it goes then binds to the LAC repressor, your LAC repressor will get removed. Now your T7 RNA polymerase will get expressed. So once your T7 RNA polymerase will get expressed, your T7 RNA polymerase will go and bind to the T7 promoter, and it will start transcribing your gene of interest. So this type of control, why is such a type of control necessary? Because because the reason is that you do not want the bacteria to continuously express your protein, right? And also there are certain type of protein, if they are expressed in very high quantity in the bacteria, then that becomes toxic to the bacteria. Okay, so that is why we follow this IPTG induction method. That is, when a bacteria has reached a particular growth, so in the log phase, when it has reached the log phase, we add the IPTG, and then the protein expression is induced. Is this clear now? Little bit.
Yes, sir.
Okay, so now let's understand this figure. So that is what I was explaining. You have the E. coli genome. So the E. coli genome, you have the LACI gene and you have the DE3 gene. So this is called the BL21 DE3 strain of E. coli. Okay. So if you work in any structural biology lab, you will be working with these type of strains. Okay, and then so these are very routinely used. So this you have this LACI gene. Your LACI gene is encoding the LAC repressor, and your LAC repressor is going and binding to the LAC operator. And this E. coli RNA polymerase is now no longer able to synthesize your T7 RNA polymerase. Now, what happens is that there is always some basal transcription. Okay, some leaky expression always takes place. So what is done? There is some strains which have this P-lyse S plasmid as well. So what this P-lyse S plasmid does, it produces this T7 lysozyme gene, and then this T7 lysozyme gene will code for T7 lysozyme. And if there is any basal expression of T7 RNA polymerase, it will digest this RNA polymerase. Okay, it will digest it. Okay, but so your pet vector also has this LACI gene, and this LACI gene will also code for LAC repressor, and it will not allow the. So even if there is any basal expression of T7 RNA polymerase, this your gene of interest will not get expressed because your repressor is binding to your LAC operator. Now, once you add this IPTG, your repressor gets removed, your T7 RNA polymerase getting synthesized, and then this T7 RNA polymerase will go and bind to the T7 promoter, and the expression of the target gene will start. Okay. So actually, there are different strains of this expression host. You have this BL21D3 strain. Then there is another strain which is called the BL21D3 P-lyse S. So P-lyse S means what? This strain has this bacterial genome is there. You have engineered this bacterial genome, and along with this, it also has this plasmid, P-lyse S plasmid. So that if there is any basal level of expression of T7 RNA polymerase, then that can also be destroyed or degraded by the lysozyme enzyme. Okay, so there are different types of host, and you have to actually optimize that which host is working in your case, or cloning your gene of interest in which host is giving you better yield of protein. So lot of optimization is required. Okay, so you cannot actually say that this host will work and this will not work. You have to check and see. Okay, so the E. coli genome in DE3 lysogen contains the T7 RNA polymerase gene under the control of LAC UB5 promoter. So this is your LAC UB5 promoter. This one. This is called the LAC UB5 promoter. And this T7 RNA polymerase gene is under this control of LAC UB5 promoter. So the LAC repressor binds to the LAC operator, preventing the transcription of T7 RNA polymerase in the absence of IPDG. So in the absence of IPTG, your LAC repressor will bind and it will not allow the transcription of T7 RNA polymerase. Now, once this IPTG induction is done, the LAC repressor is inactivated. So your LAC repressor will get removed from the LAC operator gene, and your E. coli RNA polymerase can transcribe your T7 RNA polymerase. So once your T7 RNA polymerase is synthesized, this T7 RNA polymerase will go and bind to the T7 promoter, which is present on your plasmid. Okay, so once T7 RNA polymerase goes and bind to the T7 promoter, the gene of interest, which is present downstream to your T7 RNA polymerase, that will get transcribed. Okay. So the T7 lysozyme, so the pet vector also includes a LAC operator or promoter system. So your bacterial genome has a promoter operator system, and the pet vector, your pet vector also has this promoter operator system. Okay, so is it clear now? So this T7 lysozyme encoded by P-lysis or P-lyse plasmid is a natural inhibitor of T7 RNA polymerase. And this inhibition, it reduces leaky transcription of target gene, ensuring that expression is tightly controlled and minimized when not induced. So that is also excess. So you do not want the expression of the protein, um, uh, uh, or there should not be leaky expression of the protein, because most of these proteins become toxic to the cell. Okay, that is why there is at a particular OD you have to do the IPTG induction, and that also requires a lot of optimization, whether you want to add IPTG when the OD is 0.6 or when it is zero 0.4 or if it is 0.2. So that all things you have to optimize in the lab. So at which OD your IPTG induction is giving you more yield of protein, and those that is called the optimization of IPTG induction. So those things needs to be done. Okay. So now there are different types of promoter actually. So your expression vector can have this stack promoter, which is combination of the TRP promoter and lag promoter. Then there can be TRC promoter. Then there can be lag promoter. So different type of promoter can be present. Then there is again another type of promoter which is called the lambda PL promoter. So this lambda PL promoter is basically a bacteriophage promoter, and this is regulated by a temperature sensitive repressor CI 857. So what happens is that at low temperatures, this CI 857 represses PL driven transcription. Okay. Uh, at elevated temperature, repression is lifted and transcription proceeds. So these are the different PL vectors that are available: P hub, PPLC. So what this promoter does is that, so this PL promoter, so this is your lambda PL promoter. So this is your lambda PL promoter, okay, this one. And then this lambda PL promoter is controlled by one of the repressor. Okay, so you have the operator gene, isn't it? You will have the operator gene. So this is the repressor protein, CI857. CI857. So this promoter is a temperature sensitive promoter, which means what? That this CI857 repressor protein will be produced at low temperature. So when you have kept the bacteria at low temperature, then this CI857 will get, this repressor protein will get expressed, and it will not allow the transcription. Okay, there will be no transcription. Now once you take out the bacterial culture and you incubate it at higher temperature. So at higher temperature, what will happen? So this is temperature inducible promoter. So there are different types of promoters. These are temperature sensitive promoter, you can say. So at higher temperature, this repressor CI857. So this repressor CI857 that gets removed. That gets removed and your transcription can start. So here you are not adding IPTG, isn't it? So how are you regulating the expression of the protein? By regulating the temperature. So when you are keeping it at low temperature, your protein is not expressed. Now, once a particular, your bacteria has grown to a particular OD, you have got zero point 0.6 or zero point 0.5, then you are taking out your culture and you are placing it at higher temperature. Once you place your culture at higher temperature, then your protein will start getting expressed. So it depends on what type of vector you are using. So if it is a PET vector, then it is an IPTG inducible vector. If you have these type of vectors, these are all commercially available. Okay, so you don't have to get overwhelmed by these terms. Okay, P hub, PPLC, PKC30. So these are the vectors which are having this type of promoter, lambda PL promoter, which means that you do the induction by regulating the temperature. Okay. So are especially useful for genes that produce products toxic to E. coli. So that is what I was explaining. So if they are, you want to express the protein only when it is required. You do not want basal level of transcription of your gene of interest, because that would become toxic and your protein might go in the inclusion bodies and then it will be insoluble. Okay. So now answer this question, the promoter that is regulated by a temperature sensitive repressor. What is that? Just now we discussed
Option D.
option. Yeah, option D, lambda PL promoter. These things are clear, right?
Yes,
So if
sir.
you, okay, you understood this. Okay. So this is a temperature sensitive promoter. Okay, you will do the expression by regulating the temperature or incubation. Now expression vector containing bacteriophage T7 promoter. So that is the PET vector system that we understood, that we learned just now. So your PET vector family. So there are different types of PET vector. Okay. PET28A, PET11A. These are very routinely used. Okay. Or PET3A. these different types of PET vector and presence, and all these PET vector are actually IPTG inducible. Okay, so this PET vector has a T7 promoter. So this T7 promoter, you have the T7 promoter, and downstream to the T7 promoter, you have cloned your gene of interest. So if you want to clone your gene of interest in a PET vector, then all these PET vector has this T7 promoter upstream to your gene of interest, and this is your terminator. Okay, this is your terminator. And actually, between this promoter and gene of interest, you have an operator. This is a lac operator. Lac operator. Okay. And here you also have the LAC-I gene. Okay, so if you see this one, so you have a LAC-I gene. I'm drawing it very bad. So this is the LAC-I gene. So what happens? So this gene will code for your repressor protein. So it will code for the LAC repressor. Okay, it will code for the LAC repressor. So once you have this LAC repressor will go and bind to your operator. Okay, it will go and bind to your operator, and your T7 RNA polymerase will not be able to transcribe your gene of interest. Now, so you have this T7 RNA polymerase. This is what? This is your T7 RNA polymerase. This is your T7 RNA polymerase. So your T7 RNA polymerase cannot transcribe your gene of interest when in absence of IPTG. Now, once you add IPTG, your IPTG will go and bind to your repressor. Okay, and this repressor will be removed. This repressor will be removed, and now your T7 RNA polymerase can transcribe your gene of interest. So this is the mechanism of if your protein is if your gene of interest is cloned in a PET vector, then this is the mechanism by which your protein gets expressed in a gene in a if your gene is cloned in a PET vector. Okay, this is clear, right? So remember that PET vector are IPTG inducible vector system. Okay, so I already explained you this slide. Okay. Now, answer this question. The growth phase of bacterial culture induced with IPTG for protein production is what? So, what happens is that when you have, so you have a for example, you have a 750 ml LB media. Okay. So this is your 750 ml LB media. And now you have inoculated this with your bacterial culture. You have a bacterial culture, and you have poured this bacterial culture in this 750 ml LB media. Now, what is done is that this culture is incubated at 37 degrees Celsius, and your bacteria is first allowed to grow. Okay, so and once you incubate it at 37 degrees Celsius, what will happen? You will keep it at shaking. Okay, you place it in an incubator. The incubator will you have to set the RPM, and once you keep it at shaking, your bacteria will start growing. So for around it can take around 2 to 3 hours. So within this 2 to 3 hours, your bacteria will start replicating, right? So at each 20 minutes, your bacteria will start dividing, which is the doubling time of bacteria. And after 2 to 3 hours, you will have a OD of zero point 0.4 to zero point 0.6. So at this phase, when your OD, when the culture has reached an optical density of zero point 0.4 to zero point 0.6, then most of your bacteria is in the log phase, isn't it? Is in the log phase, which means that this bacteria is growing very rapidly. So now, once your bacteria is in the log phase, at this stage you add IPTG. Okay, because if you will all you want the expression of the protein to so if there is not sufficient bacteria in your culture, then there will be no expression of protein. Okay, so you want your bacterial culture to be in the exponential phase. Okay, because at the exponential phase you will have the maximum number of bacteria, and your bacteria is rapidly dividing. So if you have sufficient bacteria at this stage you do the IPTG induction. Okay, so the answer is log phase actually. Okay, so you see this figure, isn't it? I hope you understood this in the lecture as well. So this is the general protocol that is followed in the lab. So you have your recombinant plasmid. You have cloned your gene of interest and other stuff. You have done everything. You have this recombinant plasmid, and then what you have to do after you have got your recombinant plasmid, you have to do transformation. So you have to clone your recombinant, or you have to put your recombinant plasmid in the competent cell. You have to make the bacterial cells competent enough to take up the plasmid. So once your bacterial cells have taken up the plasmid, recombinant plasmid, then you plate it. So this is the LB agar. And you what you do is this LB agar is supplemented with a particular type of antibiotic. So this plasmid also we saw last time. This vector has different type of selectable markers, isn't it? So these are basically selectable markers are what? They have the antibiotic resistant genes. So only those bacteria which have taken up the recombinant plasmid, only they will be able to grow in this LB agar, because your LB agar has, say, for example, ampicillin resistant, or it has ampicillin antibiotic, then only those bacteria which have the ampicillin resistant gene, only they will be able to grow. So once you have got the colonies, you pick up the colonies and you inoculate in a less culture volume. Say, for example, you have inoculated a 100 ml culture, 100 ml LB media. Now you put it inside the incubator. So this is your incubator. It is maintained at 37 degrees Celsius, and you have set it at 180 RPM. So once you set it at 180 RPM and provide the particular temperature, this bacteria will grow. So then once this grows to a particular OD, 0.4 to zero point 0.6, then you add the IPTG. So this concentration is what is also one thing that you have to optimize. So once you add the IPTG, then your protein will start getting expressed, right? So all those processes will start taking place. Your repressor will get removed, and your T7 RNA polymerase will transcribe the gene and will get the protein. And once you get the protein, then after you get the protein, what you do the centrifugation, and you pellet down all the bacterial culture. So once you have the bacterial pellet, in the next step, you go for sonication. Sonication. So you rupture the bacterial cell wall, and then once you rupture the bacterial cell wall, you will get the protein. Okay, your protein will come. You will again spin it. So if it is a soluble protein, you will get it in the supernatant. But if your protein is expressed in the inclusion body, then you will get it in the pellet. Okay. So now, what are fusion proteins? So what are fusion proteins? So what happens is that, as I was explaining, that when you are expressing a particular protein, there can be two, three different outcomes. Okay, either your protein can come in the soluble fraction, or it can come in the insoluble fraction. Now, insoluble proteins are very difficult to purify. Okay, so usually what is desired is that my protein should be soluble. Okay, because if it is soluble, then the further downstream processes becomes easier. Actually, okay. So now there is one way by which you can make your protein soluble. So if you have your gene of interest, which will express your protein. Now, for example, the protein that is getting expressed is coming in your insoluble fraction. Then what you can do is you can add a fusion tag to your gene of interest. So this is your fusion tag. So there are different types of this. What are these fusion tags? It can be a GST tag. It can be a sumo protein tag. So what these GST tag, this sumo protein tag, what these does is that they synthesize different types of protein, GST protein or sumo protein. And because this sumo protein and GST protein, they are soluble, your protein that you have that is getting synthesized from your gene of interest, that also gets soluble. Okay, so if you want to convert your insoluble protein to soluble protein, then what you can do, you can add a fusion tag. You can add a fusion tag to your gene of interest, so that your insoluble protein will come in the soluble fraction. Okay. So then another type of fusion. So there can be different types of fusion tags. Okay. So if you want the insoluble to soluble, then you can add this GST tag or sumo tag. You can clone this GST tag, sumo tag to your gene of interest. Then this is one method. Another method is that if you want to purify your protein, then what you can do, you can add a 6 histidine tag. This is the 6x histidine tag to your gene of interest. You can clone your gene of interest uh, having this. So now the 6x histidine tag can be either cloned in the N-terminal region, or you can also clone it in the C-terminal region. So why is this done? So for purification of your protein. So once you have added this 6-histidine tag to your gene of interest, then the protein that will get synthesized, the protein that you have synthesized, that will also have this histidine tag. Now this histidine tag can be used for nickel NTA purification. Okay, affinity chromatography if you want to do, then this will be useful. And then uh, uh, another why you need fusion protein is that to protect the target protein from proteolysis in prokaryotic host. This is one, to improve the solubility of the target protein. And the third is that to isolate the target protein. So that is what I was explaining. So you can add histidine tag if you want to isolate the target protein, or joining the target protein to topogenic signals may allow the fusion protein to be directed to particular cellular compartment. Okay, so if you add a nuclear local or any signal peptide to your gene of interest, and you want your gene of protein to be localized to a particular cellular compartment, then you can add a particular fusion tag that will make your protein go to that particular compartment. So these are the different reasons why the gene of interest is cloned along with a fusion tag. So first is, if you want to make your protein soluble, then you can add this sumo tag, or you can add this GST tag. Then there are other types of different types of tags are there which you can use. Then if you want if you want to purify your protein, then you can add this 6s histidine tag, or uh, or for even if you add GST tag, then you can use for go for GST purification, column purification. Okay, and there is that you want to protect the target protein from proteolysis, then you can add some carrier sequences. Okay, now what will happen is that you want only your protein of interest to be expressed. But if you have added a fusion tag, then what will happen? Your protein, you have got your protein. Now, along with your protein, there will also be the your fusion tag will also get expressed, right? So you will also have this GST protein expressed along with your protein, or you can have your sumo protein expressed along with your protein of interest. Now you only want this protein. You do not want this GST protein. So what is done is that there is cleavage site which is added in between your protein of interest and your GST tag, so that once you add this protease, your protease will go and and cleave this linker which is present between your protein of interest and GST, and then you can get your protein of interest. Okay, so look at this figure here. So you have your promoter. Okay, you have your promoter. You have this ATG sequence, which is the transcription start site, and then you have your gene of interest. Okay, now this is not your gene of interest. This is the carrier sequence derived from the fusion partner, and now here you have the multiple cloning sequence. So in this multiple cloning sequence, you have cloned your gene of interest. Okay, so what will happen when you are expressing the protein? When you are expressing the protein, then your gene of interest will also get expressed. Okay, will get your gene of interest, and along with your gene of interest, the carrier protein that is derived from the fusion partner, that will also get expressed. Okay, but you only need this protein. You do not need this protein. So what you have to do, you have to cleave this. So you have to add that. So there are different chemicals that can be used, or there can be different types of enzymes which can act on this linker sequence, and it can separate, or it can cleave the your um, protein from your fusion protein. Okay. So now, to obtain the polypeptide of interest in the native and biologically active form, your fusion protein must be cleaved from the remainder of the or your protein of interest must be cleaved from the remainder of the fusion protein. So both chemical and enzymatic methods have been developed to cleave peptide bonds at the joints between the sequences of target protein and carrier protein. Now there are different types of, so this is cyanogen bromide. So this is a chemical, Okay. So chemical method for cleaving your fusion protein from the a protein of interest. So cyanogen bromide, what it does, it will specifically cleave at the C terminal of the methionine. So if you have methionine, then you have say arginine, then you have say leucine, then you have phenylalanine. So once you add this cyanogen bromide, what will happen? That this is the C terminal region of the methionine. It will specifically cleave at this region, and then so what will happen? Your so if you have another fusion protein, then your fusion protein will get cleaved, and your protein that you have, the protein of your interest, that will be cleaved from your fusion protein, and you can then do another round of purification and get your protein of interest. So there are different types of chemicals. So you need to remember that what is the cleavage sequence of each of these chemicals. So cyanogen bromide will cleave after methionine. Hydroxylamine cleaves between. So if there is an asparagine and proline residue, then it will specifically cleave between this asparagine and proline residue. Then there is enterokinase, which cleaves at this sequence. So aspartate, 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 then lysine. So after this lysine, at the C terminal end of the lysine, it will cleave. Then factor XA, it cleaves after the arginine residue. Arginine residue. Alpha thrombin cleaves between the arginine and glycine residue. Then trypsin cleaves at the C terminal region between arginine. So at the C terminal of arginine or C terminal of lysine, it will cause a cleavage. Then subtilisin cleaves after at the C terminal end of arginine. So you need to remember so these are different enzymes: trypsin, subtilisin, alpha thrombin, factor XA. These are different enzymatic cleavage methods, and the cyanogen bromide, hydroxylamine, these are your chemical methods of cleavage of fusion proteins. Okay. So that is what I was explaining. So you see here, so this is your fusion protein, MALI. So this is your fusion protein, and what how you have synthesized this construct. So after this fusion protein, you have added this sequence. Okay, that will code for this isoleucine, this glutamic acid, glycine, arginine, and so this is the sequence for that will be recognized by this factor X, and then you have this target protein. So once you add this factor XA, then what will it do? It will cleave after this arginine. Now, once it is cleaved, you have separated your target protein from your fusion protein. Okay, so now this can be removed by affinity chromatography on amylose columns. So in the amylose columns, you have the resin which can specifically bind to your MALI. This is one protein. Okay, this is maltose protein. So this will bind to your amylose column, and your protein of interest will get eluted, while this tag will remain bound to your column. Is it clear? Now, another method of this is this his tag. So, what is done is this: you have added this his tag at the C. This is the N terminal region of the protein. So, at the N terminal region, you have added a his tag, and after that, there is this sequence. There is this thrombin cleavage site, and then there is then you have your protein. So, once you add thrombin, what will it do? It will recognize this thrombin cleavage site, and then it will cause a cut. It will cause a cut. Now, once it has caused a cut, then you will get this 6 histidine tag will be cut from your protein of interest. Now, when you load this protein in the column, then what will happen? So now, because this is a nickel NTA column, so your nickel NTA column will bind to your histidine, to your cleaved histidine tag, and your protein will get eluted. Okay, now because your protein does not have this histidine tag, you have cleaved it with thrombin, you will get your protein in the flow through. You will get your protein in the flow through. Are these points clear? So this is the construct if you see this, look at this figure, you will understand. So this is the construct that you have designed. You have your his tag protein, then there is a thrombin. So this is the his tag. This is your his tag. Okay, then you have the thrombin cleavage site. This is your thrombin cleavage site. And then you have your gene of interest. Gene of interest. Okay, gene of interest. Now, when this protein will get expressed, what will you get? You will get this histidine tag. So there will be histidine protein. There will be 6 histidine polypeptide that will be generated. Then there will be a thrombin cleavage site. So this is this LVPAGS, which is leucine, valine, proline, alanine, glycine, and serine. You have this particular peptide that will be synthesized. Okay. And then you will have your protein, that is whatever protein the gene of interest, accordingly that protein will get generated. Now, once you add this thrombin, this will get cut. This thrombin will cut this sequence. And now what you want to do, then you have to do again a purification so that you can separate this histidine tag from your protein of interest. So once you load it in the nickel NTA column, your histidine tag will remain bound to your column, but your protein will get eluted, and now you can use this protein and do further. You can go for further steps. Okay. So this is one. Now there can be now what if you have to use yeast as an expression host. So for different hosts, you have different vectors. Okay, and each of these vectors have different promoter. So actually you have to there are a few promoters that you have to remember. So now why do you need yeast as an expression host? Why can't you use bacteria for expression of all the different proteins? Because for example, you want to express a human protein. Okay. Now this human protein has a lot of post-translational modification. The protein has some glycosylated moieties, or it has some carbohydrate moieties attached to the protein. Then in that case, if you are expressing it in a bacteria, then your bacteria will does not have has the machinery for adding all these different moieties. So you need to express your protein in an yeast expression host. And it also enables proper folding. So as I was explaining that if it is a human protein and you are wanted, because human is an what? It is an eukaryote. Okay. And you are expressing it in a prokaryote. Then often it happens that the protein gets misfolded. So, but if you are expressing it in an yeast, then it can happen that yes, your protein gets folded because it has the machinery to convert to get a properly folded protein. So and also so high protein yield, then secretion capability, scalable, cost effective, less contamination risk. So that is a yeah, if you eukaryotic expression system can be used for expressing a eukaryotic protein. Okay. Now there are different types of yeast strain. One is this non-methylotrophic yeast and another is methylotrophic yeast. So what is non-methylotrophic yeast? Non-methylotrophic means that cannot utilize one carbon compound such as methanol. So if it is a non-methylotrophic yeast, then you need other carbon sources in the media like glucose, lactose, maltose, starch, alkane. So if you are culturing this type of yeast like Saccharomyces cerevisiae, or this is K lactis, Y lipolytica, so these are non-methylotropic yeast means you have to provide all these different nutrients in the media glucose. You cannot just culture it in methanol. Okay. But if it is a methylotropic yeast, then you can only use methanol as the carbon and energy source. Okay, so the examples of methylotropic yeast are what? This is Pichia pastoris. This is very commonly used. Pichia pastoris, Pichia angosta. So these are some examples. So if you are culturing these type of yeast, then you can only add methanol in the media, culture media. You do not need these different glucose, lactose, all these things you is not required. Well, if you are culturing non-methylotropic yeast, then you need to add all these nutrients in your media. Okay, glucose, lactose. So remember the name of few of these methylotropic and non-methylotropic yeast. Okay, now how do you select recombinant yeast? So for transformation, we learned in the last class that there is lithium acetate treatment. Then there is a spheroplast method for transformation, right? Then there is electroporation. Now there are selection markers which you can use. So if you want to screen that which is your recombinant yeast and which is your non-recombinant yeast. Recombinant yeast means what? Your yeast will have your recombinant plasmid. So that will be called the recombinant yeast. So how will you screen for it? So the plasmid vector will have these selectable markers. URA3 or leucin2, TRP1, HIS4. This type of if these type of selectable markers are present, then what does it mean? So you need to add this enzymes in the media. Only then your yeast will grow. If you the yeast which has taken up the recombinant plasmid, they will be able to grow. They will be able to grow only in this URA3, while those that have not taken up, they will not able to grow. Okay. Then there are antibiotic resistance markers also that you can use for selecting which is the recombinant yeast. Now, production of protein in yeast, how you can produce. So yeast can either cause cytoplasmic expression, or the protein can be secreted in the media. So the cytoplasmic expression means what? So high protein expression level, however, protein recovery is difficult. So if your yeast is expressing the protein in the cytoplasm, it means that you have to lyse the yeast. You have to lyse the yeast. Now, yeast lysis is difficult because it has a cell wall. So you need to treat it with several enzymes, zymolyase and all this. But if it is a secretory protein, if your yeast is secreting the recombinant protein, then you can directly obtain it in the media. So the media in which you are culturing the yeast, that will have your recombinant protein. So now you can filter the media using a 0.2 micron filter, and then in the media only you will have your recombinant protein. Okay. So proteins that are tagged with signal peptides like alpha mating factor, alpha emit MAF, this is called signal peptide. So then this type of protein, if these tags are present, then that will direct the protein to go into the secretory pathway, and the protein recovery becomes easier. So so proteins which are having cytoplasmic expression, they are difficult to be recover. The recovery of such proteins are difficult, while proteins which are secreted in the media, then such type of proteins are can be recovered easily. Uh, Okay, so I forgot to add the animation. So yeah, so why do we use eukaryotic expression system like yeast? So we are using yeast because there are certain types of proteins which have usually human proteins or eukaryotic proteins. They have several post-translational modification, and that is why if we want to express such type of protein, we usually go for expression of such type of protein in a eukaryotic host. Okay, so the answer is because eukaryotic hosts have all these different enzymes like N-glycosylation. If it has to perform O-glycosylation, then all these enzymes are not present in prokaryotes. They are only present in eukaryotes. So that is why you have to express such type of proteins in a eukaryotic host system like yeast, or in a insect cell line, or in a mammalian cell line. Okay, and also your protein is properly folded if you are and the yield of protein also sometimes increases if you are expressing it in a eukaryotic expression system. Okay. You can read this. This was discussed in the lecture as well. Now, protein expression is in yeast. So, for bacteria, we go for the IPDG induction. So, we allow the culture to grow, and then once it has reached OD 0.4 to 0.6, then we add IPTG, and the IPTG induction is carried out for. So that also needs optimization. You can for some proteins. The expression is enough for if you allow the IPTG induction for 2 to 3 hours, then your protein is expressed in a good amount of quantity. But while some proteins require expression for a very long time. So after IPTG induction, you have to incubate the culture for 16 hours or 18 hours. Only then your protein is expressed in sufficient quantity. But for yeast, what is the chemical that you have to add? So you have to add methanol. Okay. Typically, 1% volume by volume is added twice daily for expression of the protein. To induce expression of the protein, what is used in case of yeast? Methanol. So in case of bacteria, if it is a if your gene of interest is cloned in a pet vector, what do you have to add? You have to add IPTG. So remember this point. But for yeast, you need to add 1% volume by volume methanol. Okay. So if it is a 750 ml culture, then how much ethanol do we have to add? So 1% of 750. So you have to add 7.5 ml methanol. And you have to and this yeast, why yeast is the doubling time of yeast is less, right? It is less. So yeast doubles in I think it depends on the strain actually. Where the Pichia pastor is required has a different doubling time. But if it is another yeast strain, it has a different doubling time. So you have to know what is the doubling time. So that is why for culturing yeast, it is kept as typically 28 degrees Celsius. For bacteria, we keep it at 37 degrees Celsius. For yeast, we keep it at 28 degrees Celsius with shaking for 2 days. You need to allow the culture to grow for 2 days. While for bacteria, growing it for 2 to 3 hours is enough. 2 to 3 hours is enough for making your bacteria go to the log phase. Okay. Rest of the steps are same. So once the ODA has reached 5 to 7, then you will add methanol and you will induce the culture to produce protein. Okay. Then once your protein is expressed, you will go for the sonication or you will treat it with enzyme. And then you can run it on gel and check how much protein has expressed. So the type of promoter used in constitutive expression system is what? So you have two types of expression system. One is this constitutive expression system and another is inducible expression. So constitutive expression is what? There are some housekeeping genes. So housekeeping genes are what? They will be expressed all the time. Okay, this is called the constitutive expression. So these promoter belongs to the housekeeping gene, and as a result, the expression is non-inducible. The protein production starts with the growth of the cell, and as a result, is proportional to the cell mass. But if it is an inducible expression, then what will happen? The protein will only get expressed when you are adding that inducer in your culture. Either it is IPTG or it is methanol in case of yeast. So only when the chemical is added, only at that time your protein will get expressed. So constitutive expression system has housekeeping gene promoter. Okay. So why do you need to go for protein purification? So this is the reason actually, because when you are culturing the bacteria, okay, there are some housekeeping genes so those housekeeping genes will always express the proteins which are required for the growth of the bacteria, right? So there will be different proteins that will be produced. So these are the proteins that is required for the survival of the bacteria or growth of the bacteria. And when you add IPTG, then you will have your recombinant protein as well. Okay. So you need to you only want this recombinant protein. You do not want all these other proteins which the bacteria has expressed. That is why you have to go for different purification methods, chromatographic purification methods, which we will see in lecture number 7. Okay. So that is what constitutive expression system means. There are certain proteins which are always expressed. Even when you don't add the inducer, some proteins are always expressed which are required for division of the bacteria, say for growth of the bacteria, different biochemical the proteins which are involved in biochemical reaction, those proteins will always get expressed. Okay, so those are called the constitutive expression. Okay, and those are called the housekeeping genes. Okay, now insect cell line as expression system. So again, if it is a very complex protein, so for example, if it is a recombinant antibody, okay, if you want to express a recombinant antibody or a therapeutic antibody, so there are different type of therapeutic antibodies which are commercially available, like this. These different map proteins, trastuzumab, or there is this different types of map like these proteins are there, therapeutic proteins are there. So if you want to uh, express such type of therapeutic proteins, okay, all these maps, different type of map, then you cannot express it in a bacterial host, or you cannot express it in a yeast host cell. You have to go for expression in an insect cell line, or you have to go for expression in a mammalian cell line. Okay, because these antibody have a very higher have a complex higher order structure, and they have different modifications. So such type of modifications you will only get when you are expressing the protein in either an insect cell line or in a mammalian cell line. Okay, so because insect cell lines, they will even better mimic your complex eukaryotic system. So if you want to express any complex type of protein, so as I was giving the example of antibody, so if such type of proteins you want to express, then you can go for insect cell line. So higher protein yield quality compared to yeast will be better, and they will be more scalable. The yield of protein will go high, and you can use it for therapeutic purposes. You can commercially, if you want that to sell that therapeutic protein as a commercially, then you have to go for this system. Now, this baculo for insect cell line expression, you need baculovirus vectors. Okay, these baculoviruses are the viruses which infect insect cell lines. So what type of cell line is used? So this is SF9, SF9, SF20, 21. These are insect cell line. Okay, these are insect cell line. So you have to infect this insect cell line with baculovirus. Okay, and your baculovirus will has your gene of interest. So inside this baculovirus, you have added your gene of interest, and when this baculovirus will infect this insect cell line, then by the process of homologous recombination, your gene of interest will be transfected or will recombine with the genome of the insect cell line, and then then this protein will get expressed, and you can do the further purification and obtain the high yield of the protein. Okay, so remember this SF9, SF21, these are the insect cell line. And for you do not have to go into very detail because you cannot remember the name of all the promoters that are there in baculovirus and there. But remember that this for infecting the insect cell line, you need baculovirus vectors. Okay. So bacterial in this baculovirus vectors, you have a baculovirus, you have packed your gene of interest. You have cloned your gene of interest. Okay. So now there is again another protocol for culturing insect cell lines. Okay, so you have to maintain the insect cell line in suitable media. So for culturing insect cell line, so for bacteria you need LB media. For yeast you will need some different type of media. Okay, so if it is a methylotrophic bacteria, you have to add methanol. If it is a non-methylotrophic bacteria, you need to add different nutrients. Now when you are culturing insect cells, you will need another type of media. So you need to provide fetal bovine serum. You have to add all these components in the media. Okay, and now for the insect cells are they are as eukaryotic cells have the doubling time is lesser compared to prokaryotic cells. So you have to need to culture it for around 3 days, 2 to 3 days. You have to culture. You have to allow the cells to grow, and then yeah, you have to harvest. So the remaining steps are same. You will induce it, and then you will harvest it and run an SDS page, and you will check how much protein is expressed. Okay. Now then there are mammalian cell expression system. So mammalian cell expression system, when will it be used? So if you need to produce heterologous protein with full complement of post-translational modification. So if you need to produce some other complex proteins, so some human proteins which are very which have a very high the structure of such proteins are they have a very intricate structure, those proteins, and which is specific to humans, then you have to express such type of protein in a mammalian host system. So commercially, about half of the commercially available therapeutic proteins are produced in mammalian cells. So the challenge of mammalian cell culture is what they're very difficult to maintain, and there is a lot of problem of contamination. So maintaining mammalian cells are very is tricky, and you need to be very careful when you are handling mammalian cells. Okay, so because it easily gets contaminated with animal viruses. Now these are some of the cell lines which you have to remember. So there you can depending on the protein you want to express, you can use different cell line. It can be a CD1 cell line, which is a kid, which has kidney origin. COS7 cell line have fibroblast origin. 3T3 cell line, these are again fibroblast origin. CHO, this is Chinese hamster ovary cell line. So ovary cell line. Then this macrophage cell line, HeLa cell line. So if you want to culture some cancer cell, you want to see the expression of protein in a cancer cell, how the protein behaves, then you can go for these different cell lines are present. Okay, so you need to remember the origin of these different cell lines. CB1 is kidney, HEC is kidney, BHK is kidney. So you have to remember. So the cell line originating from ovary cells is what? So it is CHO cell lines. CHO is basically Chinese hamster ovary cell line. So from the hamster is an animal. From there you have isolated the ovary cells, and that is called your CHO cell line. Okay, the type of cell used in 3T3. So these things you need to remember, isn't it? So this is fibroblast origin. These are fibroblast cells basically. Okay. Now mammalian expression system has two type of expression. One is transient expression and another is permanent expression. So in transient expression, what is done? The plasmid that you have transfected. Okay. So for example, this is your mammalian cell. This is your mammalian cell. It will have its own nuclei, and along with it, you have transfected this mammalian cell with your plasmid, having your gene of interest. Okay, so this is transient expression, which means that your gene of interest has not recombined with your within the host chromosome. But in permanent expression, what happens? So if this is your mammalian cell, then and this is the genome of the mammalian cell, then this plasmid that you have transfected in the mammalian cell that gets integrated within the genome of the host cell. So it gets integrated within the genome of the host cell. So what will happen? So if it gets integrated within the genome of the host cell, then whenever this human cell will divide. This integrated the gene that it has integrated within the host chromosome that will also divide, and uh, and the gene will also get expressed because this has been cloned in a promoter. So during transcription, this will also get transcribed. So this is permanent expression where the gene gets integrated into the host genome. And it results in permanent cell line that stably expresses the protein across multiple generation. While if it is a transient expression, then after 2 to 3 passages, you have to again so it gets diluted. You have to again transfect your plasmid into the mammalian cell. Okay. So what is the advantage of transient expression in mammalian cells? So when do we go for transient expression and when do we go for permanent expression? So transient expression means if you just want to screen for some cDNA, okay, cDNA, or you just want to see whether the gene of interest that you have cloned, whether it is able to express your protein only or whether so if you just want to identify or whether you want to say yes my the construct that I have developed that is able to express my protein and that protein is getting expressed on my cell surface, then you can go for transient expression just for checking. But okay, but if you want that cell to continuously producing that particular protein, then you have to go for permanent expression. So transient expression it will not give you long-term expression, isn't it? So this is wrong. Permanent integration into the genome, no. But there is permanent integration into the genome takes place in case of permanent expression. Okay, permanent expression, high transfection efficiency, high expression level for a short time. So there will be high expression level for a short time. For 72 hours, there will be high expression. But after that, once this cell starts dividing, this plasmid will no longer be there in the divided in the cell that has divided. Okay. So you need to again transfect it. Transfect the cell with your gene of interest plasmid. Okay. So you can read this. So the expression, transient expression, the expression is high, but for short period of time. Okay. Okay. Now, what is the method for procedure for transient expression? So again, the method is the same. You have to first of all, what if you have done any mammalian, if you have worked in any lab, then you must be knowing that how to do this cell culture, right? So you have to take a particular type of cell, and then you have to seed the cells, and then you have to add the media. So there are different types of media like DMEM media, RPMI media. Then you have to provide the new this serum, fetal bovine serum, and you have to add the antibiotics, and you need to allow the cell to grow. You have to culture the cell. Now, once the cells have reached a particular confluency, then you need to passage it. Need to give passages, and then again you have to grow the cell. And once you have got the appropriate number of cells, then you can what you do you have to generate the construct. So your construct will have your gene of interest, and then you add the construct in the media, and you have to do the transfection. So we learned about the different methods of transfection by liposomal method, or you have to add lipofactamine, and your construct will get inside the mammalian cell. Now you can add the antibiotic and you can screen for the cells which have taken up the particular construct. Okay, then you centrifuge it, you take the cells, you lyse the cells, and then you get the cell lysate. Now you run the cell lysate on a gel, and then you can do Western blotting to check the expression of the protein. So these are the steps. So these things are actually you will understand better if you work in the lab and if you do the if you have the chance to work in a cell culture lab, you can understand these things better. Okay, rather than me making you understand these things are something that uh, that you will understand better when you are working in a lab. So how to do this transfection. So these are different protocols, but you will not be able to remember the protocol unless you do them on your own. Okay. Now for what is the procedure for permanent expression? So again, you have this cells, you are growing these cells on different, there are this T25 flask where you grow them and then you add the plasmid. And once you have the transfected the plasmid means you will do some this usually this lipofactamine based transfection is done. So your cells will take up the your plasmid will enter inside the mammalian cell and then you have to select, you have to do some selection that because not all your cells all the so if you are having 1,000 cells, so if you are having 1,000 mammalian cells, not all the cells will take up the plasmid. Okay, there will be some cells which have which will in which your plasmid will not which where your plasmid will not be introduced. Okay, will not take up the plasmid. So once you add this antibiotic, then in presence of the antibiotic, only those cells which are having the which have taken up the recombinant plasmid, only they will be able to grow. Now, what is done then? This cloning rings are added. So once why these cloning rings are added is if you want. Now, actually, what is done is that this you have to check that which of this clone, which of this colony is able to express your protein of interest. Okay, so this cloning rings are added so that this this mammalian cell can grow separately and you can isolate this colony. You can isolate not colony. This is you can isolate this mammalian cell and then you can grow it separately and then you can lyse the cell and check what is the expression of protein in this clone, what is the expression of protein in this clone, in this clone. So you need to check that which is the clone which is giving me the maximum yield of protein. And once you have got that, yes, this is the mammalian cell which is giving the maximum yield of protein, then you can culture it separately and you can maintain it then. Okay, so this basically this why these cloning rings are added, they are added so that you can separate out this mammalian cell by adding trypsin and EDTA. So once you add this trypsin and EDTA, yeah, your this extracellular matrix proteins that are there, they can be they are they those get you can remove it. You can so if it is an adherent cell culture or adherent cell, then between the substratum, between the flask, T25 flask and your cell, these different integrin proteins or these extracellular matrix proteins are expressed so that they can remain attached to the substrate. So once you add this trypsin and EDTA, this cells, these cells can be removed from the surface and then you can again subculture it. You will subculture it separately in a 6-well plate or in a 24-well plate, and then you will check the expression of each of this mammalian cell, and then you can maintain the particular cell which will have the permanent expression. Okay, so if you are culturing it for 2 passages, 3 passages, 5 passages, 10 passages, even then your protein is getting expressed, then you can say that it is permanent expression. While if you are seeing that after 2 passages, my expression, protein expression has decreased or there is no expression of the protein, then it will say that this is temporary transient expression. But even after culturing it for several times, if your protein is getting expressed, then it is called a permanent expression. Okay, so this is all the, so you can read all this, but the main concept you need to understand that what is permanent expression, what is transient expression. Okay. So the transform and stable transformants are selected in a permanent expression system by using what? So you were using the antibiotics, right? So answer is antibiotics or auxotrophic factors. So once you add this antibiotic, so basically the plasmid where you have your clone gene of interest, those have different selectable markers. So those selectable markers they can be your antibiotic resistant genes, or they can be these oxotrophic factors like URA3 or all these that we were discussing. So that will ensure that only those cells are able to survive which have taken up this recombinant plasmid. Okay, so that is the basic concept. Now there are different methods, like different strains have different oxotrophic factors. You can go into the detail of all those which has what type of oxotrophic factor. Answer is option D. Okay. Now purpose of using a selection marker in permanent expression system is what? So why you are using a selection marker? So just now we discussed this. So we are using it for identifying the successful transfection. Okay, because if successful transfection has taken place, then that will mean that my recombinant plasmid has entered into my host cell. So once this recombinant plasmid has entered into my host cell, then this recombinant plasmid will also have some antibiotic resistant genes. So only those cells which are having those recombinant plasmid, only they will be able to grow in the media which is supplemented with that antibiotic, while other cells will die. Okay. So answer is identify successful transfection. Now then there is another this tetracycline inducible expression system. So this is the last topic of today. Okay, so what is tetracycline inducible expression system? So in bacteria, we learned about IPTG induction. Okay. In yeast, we learned about the methanol induction. So we add methanol, 1% methanol every 2 days. Okay. For expression of the protein. Now in mammals, if you are expressing a protein in a mammal, in a mammalian host cell, then also you need some chemical, or you need some chemical so that you can regulate when your protein will be expressed. Okay, so in mammals, what you have this tetracycline inducible expression system. So there are different types of vectors that are available. So for bacteria, you were using the PET vector. Okay. For yeast, you have different vectors, which is the this methyl oxotrophic strain. There are different type of vector. I do not remember the name. You can go through the lecture. So there is different type of vector. Then again, if it is this mammalian host cell, and if you are for mammalian host cell, you have different types of promoter, and you have different types of vector. So it can have SMV 40, simian virus 40 promoter. So it will have different vector. Okay. So different vector will have this different genes, operator genes, and here so that you can control the expression of the particular protein. So this what does this tetracycline inducible expression system have? So this tetracycline inducible expression system has this tetracycline operator sequences. Okay, and then it also has this tetracycline control activator. Then there is target gene and tetracycline or doxycycline. So let's see this figure. Okay, then you will understand. So this is your this is the vector. This is the vector, mammalian vector, where you have cloned your gene of interest. So your mammalian vector has this tetracycline operator gene. Okay, and it has this promoter, and then this is your gene of interest. So what happens when tetracycline is added? When tetracycline is added to the media, then this repressor. Okay, when tetra so look at this. So in absence of tetracycline, this this TTA protein. What is this TTA protein? This is tetracycline controlled transactivator protein. So this tetracycline controlled transactivator protein binds to the tetracycline operator sequences. Now binding of the tetracycline transactivator activates the minimal promoter, initiating transcription of both target gene and potentially the TTA gene itself. Okay. This leads to a high level of expression of the target gene. But in presence of tetracycline, your gene will not get transcribed. It is the repressed state, because in presence of tetracycline, your tetracycline binds to the transcriptional transactivator, changing its shape so it can bind the TET operators. So this is the TET operator. So at this TET operator, your so this is actually, you see, this is the tetracycline transactivator protein. So this is the tetracycline transactivator protein. So when this tetracycline transactivator protein is acted upon by tetracycline, only then it can bind to the tetracycline operator. So in presence of tetracycline, your your gene will not get transcribed. Okay. And in absence of the tetracycline, your gene will get. So in presence of this tetracycline, your gene will not get transcribed. And in absence of the tetracycline, your gene will get transcribed. Okay. So note, so see this table. So in absence of tetracycline, this no tetracycline means this transcriptional transactivator will bind to the operator and there will be high expression. While with tetracycline, what is happening? This transcriptional transactivator will not bind to the operator and there will be no expression or very low expression. So this is somewhat different from the bacterial IPDG induction system that we learned. So in absence of tetracycline only there will be expression. In presence of tetracycline, there will be no expression. So when is expression taking place? When this tetracycline transactivator is able to bind to the operator. Only then transcription is taking place. But if it is not able to bind to the operator, then there will be very less expression or no expression of the protein. Okay, so this is the last question for today. The role of tetracycline in the tetracycline controlled inducible system is to block TTA binding, enhance TTA binding, degrade TTA, phosphorylate TTA. So tetracycline, when it is present, when tetracycline is present, it will inhibit the binding of, it will inhibit the binding of tetracycline activator, tetracycline transactivator to the operator. The operator. Okay, it will inhibit this. And if your tetracycline activator is not able to bind to the operator, then there will be no transcription. So what your tetracycline is doing, it is blocking TTA binding. Okay, this is this transcriptional transactivator. This transcriptional transactivator binding to the operator is blocked by tetracycline. So there will be no expression. While if your tetracycline is not present, if it is not present, then your expression can take place. Okay. Expression takes place. Okay, because your transcriptional transactivator, tetracycline transactivator can bind to your operator and your expression can start. So answer is A. Okay, so that was all for today. Okay, uh, so I think I got. Actually, there are so many concepts that are discussed, and if everything has to be covered in 2 hours, then I have to go a bit fast. So, if there are any points which you couldn't understand or which you require clarification, then you can ask me. Okay. So if there are any other points which you need more, which require more clarification, then you can directly ask me in the next class. Okay, we can go slowly and explain you each of the concept. So, uh, and also this video will be uploaded in the YouTube, so you can go through that particular section if you couldn't understand that part. So do you have any doubt in whatever was covered in today's session? So So we covered all those topics that if we want to express the protein in bacteria, what all things we have to consider. If we want to express in yeast, what are the things we have to um, consider in the mammalian cell, in insect cell, what are the promoters which are present, what are the vectors that are present if it is a mammalian host system, if it is a bacterial host system, if it is an insect cell line, what are the different vectors that are present. Okay, and what are the different inducible factors that needs to be added for expression. So for bacteria, if it is a pet vector, need to add a IPTG. If it is an yeast, we need to add methanol. If it is a mammalian, we need to add tetracycline. So tetracycline will stop the expression. And if there is no tetracycline, then expression will start. So these are some of the things which you have to remember for now. Okay. So I hope you understood the concepts and we'll meet again in the next class. Okay. Thank you for your
Thank you.
patience.